Now we're going to look at how it actually replicates. DNA replication is the vocab word for copying a DNA molecule. It is replicating itself. We call it semi-conservative. And I'll explain that when we see the picture here in a second. It's called semi-conservative. What that means is each strand of the DNA of the original parent essentially can become a new copy. It serves as a template or a model for a new strand in a daughter molecule. That gets a little more complex if you study genetics, but for our class, we're just going to keep it at this simple. Each strand becomes a template. So here's a picture of DNA. We talked last time about how it runs in that anti-parallel fashion, where what you have on one side is essentially the opposite on the other. So if you have a C on one side, you have a G on the other. T on one side, A on the other, and it runs both ways. It's called semi-conservative because the original strand of DNA, both of those strands, are used as a template for us to form two new strands. Thank you for telling me you're here. If it makes you feel better, I don't actually look at when you say you're here. I look at who logged in, how long you logged in. I do, well, that's not true. I do read when you say you're here. That's not true, so keep doing it. What I look at for attendance, however, on Zoom is if you are answering my question. If you're, which again doesn't apply to any of you because you sit in the seat, but when you're on Zoom, you don't answer the question 10 minutes in or 30 minutes in or whatever. That kind of tells us you turned it on and then, you know, went to your job or went downstairs or whatever. So anyway, with that, this kind of confused me for a while. I didn't understand why it was called semi-conservative. I didn't get it. And so if we have our DNA strand, I don't have different colors of chalk. We're getting ready to learn that DNA essentially opens up. And then two new strands are formed. So it's semi-conservative. We've conserved the parent strand. If it were conservative, we would just go through and read this and then just make a brand new copy. And that's not what happens. It's not true, just run it through a machine, get a new copy of it. We have to split open the old copy, and then both of those become part of the new copy, essentially. That's why it's called semi-conservative. Maybe it doesn't confuse you that much. For some reason, when I learned it, it really just, I didn't understand why it was called semi-conservative. It's just because parts of those parent cells, or parts of the parent DNA is used to make the two new strands. It requires several steps. DNA is coiled and twisted and all over the place inside the nucleus. And so we have to actually unwind the helix. There's a special enzyme that you have to know for the exam. There are a couple enzymes in this chapter. The first is called helicase. Helicase unwinds the helix. That's that twisted shape, called a helix or a helical shape. So helicase unwinds the helix. They try to help you out there with that name. Once we're unwound, we can open up our DNA, and then you think, well, how do we know we're making exact copies? Well, if you think back to last lecture, we said DNA is anti-parallel. Whatever we have on one side, we have on the other side, all in the opposite way. So if we split this open, and we still follow our complementary base pair rules, we'd obviously end up with two exact copies. Because wherever we had an A, we'd attach a T, which is exactly what they had in the parent copy. Where we have a C, a G, a G, a C, a T, and A, so on and so forth, all the way down to the bottom of this strand. And so complementary base pairing, that pyrimidine to a purine, double ring to a single ring, allows us to make exact copies. Each daughter DNA molecule contains one old strand and one new strand. That's part of why it's semi-conservative. Each daughter strand contains one old strand and one new strand. We'll first talk about prokaryote DNA replication and then talk about us eukaryotes. Prokaryotes is pretty simple. They have a circular chromosome. The circular chromosome replicates itself in a loop. We begin replication and it moves around the circle until we're complete and we have 2 new chromosomes. This process occurs relatively quickly. Bacteria can reproduce relatively quickly and they can replicate their chromosome relatively quickly, so much that they're not necessarily tied to one another. You can replicate your DNA separately than replicating yourself. And so they essentially are replicating DNA before they even begin to divide. And that's why bacteria, or another reason bacteria are so successful, because they're so quickly replicating that DNA that once they start dividing, they have plenty to go around. So here's a picture of that. It's just a circle chromosome at the top. We begin replication and it moves around in a circle. And then you're done. And that's bacteriotic replication. Because remember, most bacteria only have one chromosome. Maybe not even that many strands of nucleotides, really, as opposed to all the different kinds that we have. So with eukaryotes, because we have so much DNA that has to be replicated, if we just started at one spot and worked all the way through the chromosomes, we'd never finish. And so what has to happen is replication begins at many different positions along the chromosome, and they work outward until they meet each other. And so this process that we're getting ready to discuss of how DNA replicates, it's happening in multiple spots all along the DNA, all along the chromosome. And so our DNA has to unwind. So what enzyme is unwinding the helix? Do you remember? Helicase. Remember, enzymes end in ase, so that part we know. We have to think of the first part of the name. The helicase unwinds the helix. Good job. And so DNA is unwound, and then it has to unzip. That's the word we call it when I drew it on the board like that. And I took our DNA, and I broke those hydrogen bonds. So we're now unzipped. Somebody has come through and broken those hydrogen bonds. We now have those 2 templates. We know that we can form a new strand based on complementary base pairing. If we have an A, all we have to do is go in and put a T. If we have a C, all we have to go in and do is put a G. You know, cytosine, guanine, adenine, thymine, and so on. The enzyme who is responsible for building the new strand of DNA, the enzyme who is attaching the new nucleotides, is called DNA polymerase. And that's the another enzyme that you have to do. DNA polymerase. DNA polymerase is going in, and where a C is an A, it's attaching a T. Where C is a C, it's attaching a G, you know, so on. T to A, G to C. This is occurring. DNA polymerase is attaching to the chromosome in many, many different locations. And so it's happening in multiple spots and occurring bidirectionally. That means both ways. So you're starting here, and you're spreading out until you meet each other. Because remember, we have far too much DNA. If it just happened in one spot, we'd never get done. And so we have to do this in multiple spots and meet each other. Eventually they meet, they unhook, and we have 2 new daughter DNA strands, or a new daughter strand. Again, it's called semi-conservative. I repeat myself over and over like I always do, just so it sticks in your head. But It's called semi-conservative because one strand is new and one strand is old. Each new parent strand is part of the old, or I'm sorry, part of the new DNA. Let me show you what it looks like. So here's a picture. Look at the bottom. I have a pointer in here. But we have the chromosomes. Here's just a strand of DNA. DNA polymerase can attach in multiple locations and work outward in both directions. And so we would begin this process in several locations, working left, working right, 5' to 3', and then we would eventually meet each other and we'd end up with 2 new strands. And so this can happen in multiple locations and meet in the middle, as opposed to prokaryotes, which just make one single chromosome. Here's a picture to kind of show you that, so you can see that process. If here's our DNA strands, starting in that 5' to 3', we can attach DNA polymerase and it kind of just chugs along, and everywhere it sees an A, it attaches a T, sees a G, it attaches a C, and so on. There are a lot of rules for why this happens, a lot of individual enzymes and processes and fragments that we don't necessarily deal with in this class. So it is a lot more intricate than this, but we just need to know the basics. This process of replicating DNA is extremely accurate, but it is not fail-proof. It's one of the reasons that even if something asexually reproduces, it's never a perfect copy of the parent. There's always something that's a little bit different. Because every so often, every 100,000 base pairs, on average, it makes a mistake that isn't fixed. Have you ever written a final draft for one of your writing classes or a comp class, and you've read it over and over and over and over again? You turn it in and you get it back and you still missed a point because you spelled there wrong or you missed a comma or something. But you know that you've read it multiple times. How did you miss it? Well, just by the very nature of how many times you read it, sometimes your brain just kind of skips over because you know it's going to be there. That's kind of the idea behind this DNA polymerase. It's very, very good at what it does, but every so often it's going so quickly it just misses one. And so this process, it just happens. Most of the time it doesn't mean anything. There's so much in our DNA that doesn't really code for a protein. It's other stuff, or it doesn't actually code for proteins at all. It's just kind of filler there because of errors. And so it may not do anything at all. Or it could be something extremely serious. It could suddenly, the shape of your blood cell may not be correct. It is capable of identifying and correcting. We've talked about those checkpoints. Remember those back in mitosis? We had those stop sign checkpoints where you stop and make sure everything looked okay. We can kind of go through and check, but again, that's not fail-proof either. Some things are missed, and that's how there are mutations. And we've not gotten to chapter 13 yet, but when we think about mutations, they're not always negative. Sometimes mutations add variations that are good for a population. It's very, very good for sexually reproducing organisms to be different. Your survivability and your fitness skyrockets if you're all different. If you're all exactly the same, suddenly you're all susceptible to everything that's the same. You all have the same water requirements, the same nutrient requirements, the same susceptibility to a virus, the same weird mutation. And so it's very good that if for everyone to be different. So DNA polymerase, in a small, small way, provides some of this variation. And it's one of the reasons that eukaryotes have been so successful. DNA polymerase grows from 5' to 3'. Remember, I said the direction, the 5', if we could draw that pentose sugar, deoxyribose, if you ever take chemistry, you'll know that we can name some of these bond areas. We can name them 1', 2'. That just means location. And so 5' to 3' is just how that molecule is oriented. For most of you in this room, you will never need to know that again for the rest of your life, that it goes from 5' to 3'. but this is that foundation class for all different types of sciences. And some people, this is extremely important. That's why we have to care. But just remember 5' to 3', and then maybe you'll never think about it again. It's one of those things.